So 1610 is finally we're going to talk a little bit about the cap protein and how the cap protein works. So remember, we talked about at least two different ways the lac operon is regulated. One of them is by the LAC-I repressor, and the other is by the cap protein, so catabolite activating protein. So I want to diagram the operon again. So LAC-I is upstream, not part of the operon. Then we have the promoter region, and I'm going to split the promoter into the RNA polymerase binding site and the cap binding site. And I'm going to draw the cap binding site with a couple DNA sequences that the cap protein likes to bind to. Then we have the operator, LAC-O, and then Z, Y, and A. Now, cap protein here, now the cap protein is going to look something like this. Okay, and it is going to bind here, and when the cap protein catabolite activating protein cap, now this is going to bind here, and when it binds there, well, it's going to help RNA polymerase bind to the promoter, so the polymerase binding region of the promoter. So it's going to greatly increase the rate of RNA polymerase binding and transcription of the operon. Okay, but it doesn't always, the cap does not always bind to that site. So it only binds to that site when glucose levels are low. And how does it know when glucose levels are low? So there's a molecule here. There's one called ATP. We know what ATP is, right? ATP is adenine ribonucleoside triphosphate. So now that can be converted into a compound called cyclic AMP. And cyclic AMP, ATP to cyclic AMP is controlled by an enzyme called adenyl cyclase. And adenyl cyclase is inhibited by high glucose levels. So when glucose levels are high, this is turned off, and as a result we have low cyclic AMP levels. If glucose levels are low, well then adenyl cyclase works and we make lots of cyclic AMP, so this would be high. So glucose low equals cyclic AMP high. Now, this is good because when glucose is low, well then if lactose is around, we want to start using lactose instead. And so this is going to be our cyclic AMP shape right here, this little blue kind of rainbow or horseshoe here. It's going to bind to cap, and then this is going to be able to bind to the cap binding site. So I should kind of diagram it like this. So before cyclic AMP binding, cap looks like this. After it binds, it looks like this, and we have the binding site now for the DNA. Okay, this is going to bind up here, and as a result, RNA polymerase is going to be recruited at a higher rate to the operon. Operon's going to be transcribed, and we're going to make those lactose catabolizing genes. Okay, and again, so we'll have the we'll assume lactose is high too, and as a result, lactose will be bound to the LAC-I repressor, and then we'll have the allosteric shift, and so the LAC-I repressor will not be bound here either. Okay, so that is how the catabolite activating protein stimulates transcription of the LAC operon when glucose levels are low and lactose levels are high. So now the question says, predict the effect of the inducibility of the LAC operon of a mutation that disrupts the function of the CRP gene. Now, okay, what is the CRP gene? Let's take a look here. Let me scroll down a little bit here. Okay, the CRP gene, CRP, which encodes the CAP protein. Okay, the CRP gene makes CAP in the CAP binding site. CAP binding site in the promoter. Okay, so if we mutate this, will the LAC operon still be inducible? LAC operon inducible. So if we add lactose, will that turn on the operon? And I would say yes. Yes, it will. Okay, why? Because we're adding lactose. Lactose is going to bind to the LAC-I repressor, and the LAC-I repressor is not going to be able to bind to the operator, so RNA polymerase is going to be able to transcribe the operon. It's just not going to be able to transcribe it at the high level it would be able to when CAP was there and CAP was bound. So RNA polymerase is still going to transcribe, it's just going to transcribe it, let's say, at a medium level instead of at a high level. So medium level, I'm going to say that down here, or moderate level, let's call it. Moderate expression, okay, when lactose is added. Okay, what about the CAP binding site? So CAP is there, the CAP protein is there, CAP binding site is mutated, sort of the same thing, you know. If lactose, we add lactose to the cell, lactose is still going to bind to the LAC-I repressor, the repressor is not going to bind to the operator. RNA polymerase is still going to be able to transcribe those, but it's not going to bind as high a rate as it would if we had a functional CAP binding site. So here I would say moderate expression again. So if both of these were working, if the CAP binding site is working and the CAP protein is being made, then we would expect to see high levels of expression of the LAC operon when, at least when glucose levels were low. I should add that the answers in the study guide for both of these cases are similar to mine, but not quite the same. The author of the study guide says that inducibility will be compromised in both cases, and well, in the first case, and reduced in the second case. Essentially, it's the same thing as what I'm saying here, where we would see moderate expression in the presence of lactose. So either answer is fine. We're not getting the high expression, the high inducibility. So the ability to induce expression of the LAC operon is slightly reduced when either the cap protein or the cap binding site is mutated.